Okay, so what we're going to learn about today is what happens when the electrical impulse reaches the very end of that neuron. So we're looking here at the point between perhaps a sensory neuron and a relay neuron, or a relay neuron and a motor neuron, or even between a motor neuron and either a muscle or a gland. Now, the junction between those two neurons, or the muscle and the gland and the neuron, is referred to as the synapse. And if you look at the diagram on the slide, here we've got a picture of the whole thing, the whole synapse. And what you can see is the very top half of the picture shows let's say, the sensory neuron, and the bottom half of the picture has the relay neuron, and the gap in between is referred to as the synaptic cleft, and that is between 20 and 30 nanometers across. So if you look at the label on the top left-hand corner, you can see that the label says end of axon of presynaptic neuron. So that's where our action potential is going to arrive. Okay, it's been propagated down the neuron, okay, and it's arrived at that point along the axon. And as soon as it reaches that point there, then we're going to trigger the release of chemicals into that synaptic cleft. Those chemicals are referred to as neurotransmitters. And so those chemicals, those neurotransmitters, are going to be produced in the very end point of that neuron. The end point is referred to as the synaptic knob, and because we are going to be creating neurotransmitters at that point there, we would expect to see a lot of mitochondria to make ATP. We would expect to see a lot of endoplasmic reticulum, so that we've got transcription, translation, or it'd be rough endoplasmic reticulum, and then we would also see a lot of Golgi apparatus. The Golgi apparatus would then produce synaptic vesicles, and each one of those little vesicles is going to contain the neurotransmitter. Those vesicles will then fuse. If you look at the diagram, you can see them fusing with that membrane on the bottom in the synaptic gap, or the synaptic cleft, and they are going to release that neurotransmitter by exocytosis. The neurotransmitter will then diffuse across the synaptic cleft or synaptic gap, and it will hit receptors on the postsynaptic membrane. Those receptors are going to be things like glycoreceptors on phospholipids, so glycolipids, or they could be glycoproteins embedded within the cell membrane. And if they recognise that neurotransmitter, that's going to then trigger a new action potential in the next cell. Okay, so there are two types of neurotransmitter that you need to be aware of. There are excitatory neurotransmitters and inhibitory neurotransmitters. Now, excitatory ones, they result in the depolarization of the next neuron, and so the impulse is therefore continued along the next neuron. An example of one of these is acetylcholine, which we're going to look at a bit later on in this video. The second type is an inhibitory neurotransmitter, and what this does is it stops depolarization of the next neuron. Okay, so it kind of, if you've got an impulse travelling along a sensory neuron, it would then stop it from travelling along the relay neuron. An example of one of these is GABA, which we find in our brain. Now, GABA is a really interesting neurotransmitter. It's there really to kind of calm everything down in the brain. So if the neurons become overexcited, it then kind of acts to stop those impulses travelling further and further and further, and it kind of just slows everything down. It's a regulatory neurotransmitter. So lots of people who don't have very much GABA often suffer from anxiety, they might suffer from sleep disorders, they might suffer from depression, schizophrenia, it might even have a role within ADHD, because everything in the brain is just kind of going on, and the impulses are travelling and travelling and travelling, and there's nothing to kind of squash them back down if they get too overexcited. So there's quite a lot of interest in drugs and GABA. There are foods that might replicate the effects of GABA, but there are also drugs that people are given who do have sleep disorders, for instance, to kind of slow things down and calm everything back down within the brain. It's a really interesting area to look at if you've got a spare few minutes in your day. So for this section, we're going to look at actually how that neurotransmitter is released and what it does when it hits the postsynaptic membrane. Now, what I would suggest you do is you have the diagrams that are attached to the Google Classroom alongside you, and then if you write alongside each diagram what's happening, so you end up with the whole process written alongside the diagrams. So the first thing that happens is that action potential is going to reach the end of the presynaptic neuron, and it triggers calcium ion channels to open. Once those calcium ion channels open, of course, calcium ions will then flood in to the synaptic knob. They cause the synaptic vesicles containing that neurotransmitter to then fuse with the presynaptic membrane and for that neurotransmitter to be released, and of course it's going to be released by exocytosis into that synaptic cleft. Once it gets into the synaptic cleft, the neurotransmitter will then diffuse across the synaptic cleft and it will bind with the complementary receptors on the postsynaptic membrane. That then triggers sodium ion channels to open along the postsynaptic membrane, which of course then starts a new action potential in that second neuron. We do still have that neurotransmitter sitting in that synaptic cleft, and that could be a problem because it could keep triggering new impulses to start in the next neuron. And so what we then have to do is we have to break down that neurotransmitter, and that happens with enzymes, and then the broken down pieces are then reabsorbed back into the synaptic knob where they can be reassembled and used again. So they're recycled. You have to know in particular what happens at the point where acetylcholine is released, and so that happens at a cholinergic synapse. Okay, and here the neurotransmitter is acetylcholine, and so what we have is once the acetylcholine has triggered the response, for instance, in a muscle, then the enzyme that's released into the synaptic cleft is acetylcholinesterase, and what that does is it breaks the acetylcholine down into choline and ethanoic acid, and it can break down 25,000 molecules every minute. They are then taken back, as I said, into the synaptic knob, and once in the synaptic knob, ATP is used from those mitochondria to recombine that choline and ethanoic acid back into acetylcholine. So the biggest role of the synapse is that it ensures that impulses are unidirectional, and that's because only the presynaptic membrane releases the neurotransmitter, and this is then detected by the postsynaptic membrane. So you can't have an impulse travelling the wrong way down a reflex arc, for instance. The other thing about synapses is that they allow us to have junctions where there's more than one synapse present, and that can work in two ways. The first way is that we have several presynaptic neurons. So if you look at the diagram here on the left, and they all feed into one postsynaptic membrane, and that means that our neurons are able to detect and respond to several impulses or several things that are happening in the environment or inside us, which send a message to one neuron, and therefore we get one response. On the right hand side we can have the opposite to this, where we have one impulse passing down a neuron, but then once it reaches the synapses at the end, it branches off into several different neurons, and therefore we can get several different responses taking place from one impulse. The final part of this topic is called summation. So if you look at this slide here on the left hand side, what we have is we have two neurons which are feeding into one. So we've basically got two presynaptic membranes and one postsynaptic membrane. And if we have an impulse travelling down A and it releases its neurotransmitter, you can see that that's not enough to stimulate the action potential in the next neuron. If we just have B, again, it's not enough to stimulate the action potential, whereas we have to have an impulse passing down both A and B to ensure that we have enough neurotransmitter to trigger the action potential in the next neuron. And so that's referred to as summation. And because we are summing A and B neurotransmitter, it's referred to as spatial summation. The one on the right hand side, we only have one presynaptic membrane and one postsynaptic membrane, but here if we just have the action potential just kind of once passing down, a small amount of neuron is released and not enough to trigger the action potential. So we have to have several impulses travelling one after the other to allow enough neurotransmitter to stimulate the next impulse, and that is referred to as temporal summation.